Dear students, welcome to the biology class. Today we are going to discuss about the most interesting part of our +2 biology, biotechnology, principles and processes. By now we have understood that DNA is the molecular basis of our inheritance. DNA is responsible for carrying characters from generation to generation. We have already proved it with Griffith's transformation experiments. Subsequently, Avery, MacLeod, McCarthy, they have also contributed. Hershey and Chase has proved that this is the genetic material. Once we are confirmed that DNA is the genetic material and it is very convenient to manipulate DNA, there is evolution in the study of biotechnology. So, let us start with biotechnology, its basic principles, and then we shall deal with the processes involved in this. So, let us start with the basic definition. We say biotechnology deals with techniques of using live organisms or enzymes from organisms to produce products and processes useful to humans. As per European Federation of Biotechnology, it is defined as the integration of natural science and organisms, cells, parts thereof and molecular analogous products and services. So, it is not a completely new thing. Since time immemorial, we have been brewing, we have been making curd, we are baking bread, which are examples of processes of biotechnology. Here we have some knowledge we use, that is from biotechnology, where we are either using complete organisms or their products to make a product which is useful for us. So, today we are proposing another form of biotechnology we refer to as modern biotechnology, where we have manipulated the genetic material or the DNA, and then we are going to have products beneficial to human beings like vaccines, medicines, improved varieties of fruits, vegetables, animals with defects to be rectified and all that. So, the word was coined by Carl Erke in 1917. Then let us come to the principles of biotechnology. There are two core techniques of modern biotechnology that are being developed in the 1970s. One of them is genetic engineering, where we have techniques to alter the nature of genetic material. Genetic material of all living organisms is DNA, and that of viruses are RNA, to introduce these into host organisms and change the phenotype of the host organism. What we mean by this is we are going to change the genetic structure of a given organism by insertion of new DNA from another plant, animal or microorganism to have a different organism. I mean, organism may look alike, but it will have some new production or it will have a phenotype changed. Its morphological aspects, its production, etc., are getting changed. Then the second aspect is bioprocess chemical engineering, that involves maintenance of sterile microbes free conditions to produce only growth of the desired microbes, eukaryotic cell in large quantities for biotechnological products like antibiotics, vaccines, hormones, medicines, etc. So, basically what we understand from these two, we have to start with an organism which is genetically modified. We say that is a recombinant one. Recombinant in the sense that its DNA will be recombined. It has got its own DNA. There will be addition of some foreign DNA into this original DNA structure, and we have a recombinant DNA structure. Then the second aspect of that is to find out that the inserted or newly added genes of another organism is expressing in the host organisms. And once we are completed with, the first process is over, and once we are confirmed that this is working, then we shall go for mass production of these, that we shall be employing bioreactors, or we shall use chemical engineering ideas, processes, whereby we shall grow in controlled conditions microorganisms or eukaryotic cells or plant or animal cells to have a specific product. Then there are 3 steps in genetic engineering. Now, we have to know what is that specific gene we are interested to insert it into a host or into a host organism. So, we have to identify that. With the development of technique, now we are confounded with technology, whereby we know the genes are located at specific sites, and we know the specific sequence of nitrogen bases of these particular genes. Once we identify where this piece of DNA lies in the chromosome, which chromosome, chromosome number what of the organism, then we are going to start manipulating it. Then first we have to identify the DNA specifically where it is located, in which chromosome it is located, then we shall try to manipulate that. Then the second aspect is introduction of the desired gene into the host. Once we identify the specific gene we are interested for, then we shall use techniques to separate it out from the particular organism, purify it, then develop it in such a way that it can be incorporated into a vector. And once we incorporate it into a vector, then we shall have a chimera, maybe a modified plasmid, maybe a modified cosmid or some other forms, whereby we can transfer it from a microorganism to another microorganism or to a human cell or any eukaryotic cell, plant or animal. So, in the process actually we have seen it is developing since 1970s. It was Arbor, Nathan and Smith in 1970 who discovered restriction endonucleases, which are referred to as the molecular seizures. I have told you we have identified a piece of DNA from a very large DNA molecule present in a chromosome. Once we identify that particular one, the most important aspect will be to cut precisely that piece of DNA, purify it so that it can be incorporated into another piece of DNA. Then it was Stanley Cohen and Boyer. They have made the first recombinant DNA in 1972. Then Paul Berg in 1970 was successful in introducing a gene of SB40 virus into a bacterium with the help of a lambda phage. Now, what are the different tools we use in this recombinant DNA technology or this cloning technology? I shall say these are 3 different ones. First comes the enzymes. We have different groups of enzymes. Once we understand these enzymes and once we have started working with these enzymes with ease, these are easily available in the market, there is rapid progress in the recombinant DNA technology. So, the most important one, as I have already told you, is restriction endonucleases, which we also refer to as the molecular seizures. These are the enzymes which cuts DNA from inside it cuts within the DNA molecule. We have exonucleases which cuts from outside. Restriction endonucleases are endonucleases which cuts DNA from within and from a specific site. The specific sites are having specific sequence of DNA or nitrogen bases, we shall say as the palindromes. Palindromes are like what we read from 5 prime end to 3 prime end in one sequence is same as the other. We shall be discussing in detail subsequently. The second type of enzymes we shall be using is polymerase. Polymerases are enzymes which are required for synthesis of another strand. Okay. So, DNA are polymers of nucleotides. These nucleotide monomers are joined by the polymerase enzymes, like DNA polymerase will join the deoxyribonucleotides forming a strand of DNA. Okay. Then we shall go to the third enzyme, which are the ligases. Like I told you, restriction endonuclease cuts, ligases are the enzymes which joins. The second enzyme we shall be dealing with is ligase. Ligases are enzymes which we use to join, like the restriction endonucleases cuts, ligases joins. When we have made nicks or cuts at certain points, want to add the new DNA strand to this DNA strand, then on both the sides they will be joined by the enzyme ligase. Then another important enzyme is phosphatase. It removes the phosphate group present in the 5 prime end of the DNA molecules. So, once it is been done, the advantage is it will not be joining with 3 prime OH anywhere with any other DNA molecule available. Another important was reverse transcriptase. When the genetic material is RNA, like we have the retroviruses, we have the virus like coronavirus, where the genetic material is RNA, then when we want to manipulate this or have experiments on this to verify, what we do is to get a DNA copy of the genetic material of the virus, and that genetic material can be done or worked with by working with reverse transcriptase. That will be a reverse of transcription process whereby RNA is synthesized from a DNA. So, here we shall see RNA will be reverse transcribed to DNA, and once we have a DNA we can manipulate with this. So, these are the examples of important enzymes we shall be using. In addition to these, we use many others. Then comes the most important thing in the recombinant DNA technology is to have a vector. A vector is something to transfer this DNA molecule we are interested to and from the host organism, from the desired gene to the vector, then from the once the vector transfers it, we get it transferred to the host organism, which will undergo transformation. So, vectors are normally we choose plasmids, we take cosmids, we take each artificial chromosome and other things. And once we use these vectors for cloning, that is to get a number of copies of the host, we want to take the number of copies of the desired gene, then we use this vector. The one of the most important vector is PBR, the plasmid vector, okay, that is named according to the discoverers of this. And then in the last we shall have the host organism that is going to be transformed. Here we shall transfer the desired gene through the vector, another vector, or through other processes to get the host organism transformed to have the recombinant DNA, and then we shall go for the manifestation of the gene we have introduced into this host organism from a foreign organism. So, I have told you already that there are three important tools we use in this recombinant DNA technology. The first one is enzymes. Let us start discussing about the enzymes. The first enzyme that we shall be using is restriction endonuclease. So, this restriction endonucleases are enzymes which cuts DNA from within. So, suppose we have a DNA molecule, this is having from 5 prime end to 3 prime end, and the complementary strand again from 5 prime end to 3 prime end, and here are the nitrogen bases. Then we shall see this is cutting from within the DNA, and that is from specific sites. The most important aspect is that these restriction endonucleases are cutting at specific sites. These are referred to as the restriction sites. So, the specific sites, the sequence of DNA nucleotides will be palindromes. These palindromes are nothing but small segments of DNA which reads same from both the ends. Like we have a sequence of nucleotides GAATTC. So, we can see this reads same from both the sides. Like suppose we take the word madam, MADAM, we read same from both the sides. Similarly, these are the palindromes, and when this restriction endonuclease is applied, we shall see it will be cutting at specific sites on both the strands. So, here we shall see this is going to cut the DNA. We have this okay. So, we see it is cutting from here like this, and then this is getting separated, and same thing happens on the other strand also. This is getting the second, this part AATTC, and this is involving the GA. So, we have other nitrogen bases present on both the sides. Now, after it is been acted upon by the restriction endonuclease, we shall see this is one strand and this is the other one. And we shall see here this is producing a protruding end. So, suppose we say this is the restriction endonuclease, we shall see this is coming out. We have a base that comes out of this, and here this can attach with a similar structure. Suppose we have a new DNA molecule to be attached to this, will be this, then this can attach here. It will be very easy once it is having a protruding end. And the important aspect is this is cutting at specific sites. And there are three different types of restriction endonucleases: type 1, type 2 and type 3. But in recombinant DNA technology, there is use of the enzyme type 2. And these are being named after the name of the bacteria from where it is been derived. Like suppose we say the restriction enzyme EcoR1. So, E comes from the bacteria Escherichia, CO comes from the species name coli, and R is the type, and 1 is the first that is been derived from this bacteria. So, this is referred to as EcoR1, and EcoR1 is having the restriction site GAATTC. By now, we have hundreds of restriction endonucleases available in the market in a very purified version. And once we are having these restriction endonucleases, we are able to manipulate the DNA molecules. So, we shall be discussing in detail, while we shall be discussing about how we go for the recombinant DNA process. Then the second is polymerase enzyme. I have already told you a little earlier that DNA is a polynucleotide. So, these are the nucleotide monomers, and these nucleotide monomers are joined by phosphodiester bond. And there are two strands. Each strand is polymerized from 5' to 3' direction. Now, we shall be having this polymerase enzyme that will allow us to synthesize a new DNA molecule on a DNA strand. It has made the system very easy after development of a technique known as the PCR, polymerase chain reaction technique, where we are using a Taq polymerase which is used to make thousands of copies of the desired gene. It is synthesized by an instrument, we say the PCR. So, this is the polymerase enzyme. Then the third enzyme we shall be discussing about the ligases. Now, suppose we have so this is originally from the bacteria. This is the one we are bringing from a foreign cell or from a cell which is not restricted to the individual cell, and then we are joining this. Now, these coats were brought about by the restriction endonucleases. Now, there is establishment of phosphodiester bond between these 5' and 3' ends, and then these 5' and 3' ends here. So, this is brought about by the enzyme ligase. Then the next enzyme is phosphatase. So, here if this is the DNA molecule, we are giving a schematic one. So, if we start from 5' and 3 prime end, we have all base OH in the 3 prime end and P in the 5 prime end. And once P and 3 prime ends are having phosphate and phosphoric acid and OH group, there is possibility that they are going to join. They may form a circular DNA, or wherever there will be possibility they will be joining. When we want to manipulate it and it is going to join, it may not be easy for us to manipulate. So, what we do is remove this phosphate from here, and if we do it, then it cannot join with the other side. So, this is how the phosphatase works. Then there is the one is reverse transcriptase. Reverse transcriptase has revolutionized the science of molecular biology. So, this brings about reverse transcription. So, suppose this is the DNA molecule. When it is a strand of this is converted to a RNA, then it is referred to as the transcription. The difference is just the nucleotides. Here we have deoxyribonucleotides, here we have ribonucleotides, and here we have a ribose sugar, and here we have a deoxyribose sugar. Dear friends, we shall continue with our discussion on the biotechnology and its principles. So, while we were considering the essential things for the recombinant DNA technology, we have already discussed about the enzymes. The next thing we shall be discussing is the cloning vectors. What is a vector? Vector is one which transfers something from one to the other. Like mosquito is a vector that will transfer the protozoa that causes malaria from human to human. Similarly, we have DNA molecules that are to be transferred from a vector to a host cell. So, we shall take a foreign DNA, incorporate it into a vector, then once it is incorporated, then that will be transferred to the host cell from where we have taken out the vector, then allow it to grow. Then what is the advantage here? We have a microorganism produced which contains a foreign DNA, and normally we say we use, there are two important vectors, one is plasmids and the second group is bacteriophages. So, when we shall come in detail to a cloning vector, the vector we use must have origin of replication. I have already given you hints here. I show you the diagram of a vector, that is PBR 322, which is having 4.3 kbps, that is 4361 base pairs. I have told you the first requirement of a vector has to be having the restriction endonuclease sites. So, from this slide you can very well see there are several restriction endonuclease sites like ECO R1, HIND 3, ECO R5, BAM H1 and then SAL1. So, if we take any of these restriction endonuclease and digest this, then we shall see this vector will be producing a cut. Suppose we say we have taken ECO R1, as I have discussed you, this will be bringing about a cut like this. Now, this plasmid is having a cut. Okay. Now, it has to have a site of origin. When we have a site of origin, it can replicate itself, and once it replicates itself, it can produce multiple copies of the same plasmid. That is most important. And then it has to have molecular markers. Suppose we are having a group of students from a particular college in a particular dress code. It is very difficult to find out to which class they actually belong to. Suppose we have +2 first year students and +2 second year students. We want to identify second year students. We ask them to put a necktie, and those who are having a tie with their dress, we can identify them to be the second year students. Similarly, so that is a mark we put on them. Like behind the jersey of sports person, a number is scribed. They identify this is the number of the jersey, like the Tendulkar's jersey or Dhoni's jersey. It is very common, you know it. So here we shall see a molecular marker will be there, and that molecular markers are normally the antibiotic resistant genes. So if that particular gene is present, that is giving resistance to that particular antibiotics. In presence of that antibiotics, this particular bacteria having the molecular marker or antibiotic resistant gene can grow over there. That is very important. Okay. And then this you can see here very well that there is a site AMP, that is the site where that will be having a gene which will provide resistance to the antibiotic ampicillin. Now, if we take a foreign DNA into this, suppose we are successful in introducing a piece of gene from a foreign cell, then we transfer this to another bacteria. Suppose this has been taken from a bacterium E. coli. We have reintroduced this chimera. This is the normal DNA of the bacterium. This is the plasmid or the vector we have taken, and now this is having this foreign DNA, and this one is having a molecular marker, that is ampicillin, the antibiotic ampicillin resistant gene. Then if it is completely entered and settled inside this bacterial cell, and this bacteria grows, this can grow in a medium having the antibiotic ampicillin. If it is not there, if this plasmid is not introduced this host cell, then we shall see this cannot grow in a culture medium having this antibiotic. So, from this actually we shall be identifying that this is the particular molecular marker present in the plasmid vector, which goes from a bacterium to a host another bacterium or maybe to a yeast cell. Like this will be continuing. So, I hope you have understood this. So, the third important thing is the cloning sites. I have already told you that if you are having the restriction endonuclease sites, as I have told you in this slide, that there are BAM-H1, SAL1, ECO-R5, HENT3. Any of these restriction endonuclease if we take, that will cut a nick over there, that will cut a piece over there, and that is the site where we can introduce the foreign DNA and get a chimera, a recombinant plasmid. And once this vector is ready, then we put this vector into the host cell and try to see whether this is a transformed cell having the foreign DNA inside this. And then we shall see the vector of these cloning genes in plants and animals, how we are going to change the plants or animals and get a recombinant plant or genetically modified plant and animal. Then we have another bacterium available having a TI plasmid. We use retroviruses, papillomavirus and adenoviruses also to carry these genes. We have already manipulated, we have made many copies of that into a host where finally we are going to get the final produce. Transformation is also brought about by other processes like microinjection, biolistic or gene guns that we shall be discussing subsequently. Well, students, we have discussed about the vector. Then we shall go to the process of recombinant DNA technology in general. So, here what we shall do is how we are going to have this process. The entire process will start with identification of genetic material, which we are going to isolate from an organism and finally transfer it to any desired organism. So, by now, by mapping technology, we have already identified where these genes are located. So, once we locate these genes and find out their sequence, this has already been done. We know that by the Human Genome Project, all the genes present in the cells of human beings are already been mapped. We know which gene is present in which chromosome, in which site, what is the sequence of nitrogen bases in this particular gene or piece of DNA. We know that genes are nothing but pieces of DNA. So, the first important thing will be how to take out this genetic material from this. So, we have techniques. We shall first treat the cell. Suppose we take a bacterial cell. The bacterial cell is having the bacterial cell wall made up of peptidoglycan. So, we have to first digest the bacterial wall, which is made of peptidoglycan, and for this we have to treat it with lysozyme. When we digest the bacterial cells with lysozyme, that will digest the bacterial cell wall, and then the bacterial DNA will be exposed. Then we shall extract this DNA. Then by cell fractionation, we shall take out this DNA. Then we shall treat this with or digest these with particularly the restriction endonucleases. Then we can make pieces of this and we shall identify to which piece it belongs to. Then take out that particular piece and start to manipulate. Then the next important thing is how we shall carry out this. We have a technique, this agarose gel electrophoresis. Using this agarose gel electrophoresis, we shall see the DNA molecules or fragments having different sizes and different charges will be separated. Okay, when we put in a buffer and apply electric field, they will be migrating, the smallest will migrate faster, and the size we have cut with different restriction endonucleases we can identify. And once we do this, then we shall take this, purify this piece of DNA purify this piece of DNA, and then this piece of DNA is been amplified. Matlab, here we do many copies of this. So, what we were doing earlier, we were introducing this into a plasmid, then the plasmid is introduced back into a bacterium. The bacterium was allowed to get into culture, and once they are coming into millions of copies of that, we were able to derive that. But now, we have the polymerase chain reaction instruments available. We use Tech polymerase, and using this polymerase chain reaction, then we can get millions of copies of the desired gene. You must be hearing about one thing, RT-PCR technique. Every day we hear it many times and with the TV. If anyone is going for a COVID-19 test, they are going for this RT-PCR, thus reverse transcriptase PCR. Means here they will take the total virus they are getting from these places where they are being incubated inside our body, then take out the RNA, and this RNA is reverse transcribed to DNA, and the DNA is reconstructed, and once it is done using the PCR technique, we have many copies of these, and once we have many copies of this, we have confirmed test of presence of COVID-19 in our body. You might be seeing in the TV also that sometimes someone is declared negative with antigen test is coming out to be positive with RT-PCR, because here we have millions of copies of the DNA that is specifically present within these COVID-19 ones. Now, once we have many copies of these available, then we shall put these to use for recombination, and then we shall go for the next step. So, we were discussing about the PCR, the polymerase chain reaction technique, whereby we get millions of copies of the DNA molecule, and once we have many copies of these, there is now the thing we have to introduce it into a vector of our interest. Now, here we shall see, I have already told you a cloning vector where we have introduced this piece of desired gene. Now, this is to be this gene once we have multiplied, this is the desired gene, suppose we say. Now, this is to be introduced inside the cloning vector, which is going to transform this host cell. Like we can use the TI plasmid of agrobacterium. Now, the TI plasmid is having this piece of DNA we are interested to get expressed in the host cell. Now, we shall infect the agrobacterium tumefaciens . This agrobacterium tumefaciens is to be infected with this DNA inside it, and it has got the ability to incorporate the foreign DNA into the host DNA. And when we have success in introducing this foreign DNA into the host DNA at specific points whereby we have a promoter, then this is going to express. And although this gene is not belonging to this particular host, but it is introduced and it is expressing, that solves the problem. That is the importance of this. Now, once this is done, this is the process of recombinant DNA technology in brief. We have to culture this in large amounts. For this, we have developed bioreactors. So, these are the culture vessels. So, if we take a very small culture vessel, we can do it at laboratory scale. If we use very large culture vessels having all control measures, we control the temperature, we control the humidity, we control the oxygen content, we control everything, everything goes in a control condition. We grow these microorganisms very happily, we give them the nutrients using which they will be growing very fast, then they will be growing and simultaneously they will be producing the particular protein we are interested for from this desired gene. Suppose this piece of gene is synthesizing human insulin or a particular protein, this produce a particular protein which is required in huge quantities. And once it is incorporated and this expresses, if we grow them in huge quantity, then we shall see that it is expressing, then we shall be take out the culture, then bring about processing of this, purify it, then that will be available in the market. So, basically this is about the recombinant DNA technology. I shall be showing you this in this slide. So, you can see this, a small piece of DNA is been taken, that we shall name it as foreign DNA. Then we have a vector, that is a plasmid, like I have told you PBR 322. It has all the features required for a vector. Then we shall treat these enzyme, treat these vectors as well as the foreign DNA to be incorporated by the same restriction endonuclease. The same restriction endonucleases will produce nicks on same site. Like suppose I say we shall digest it or treat it or incubate it with eco R1, then we shall see it will be producing nick after GAATTC is the restriction endonuclease, it will produce a nick after G on both the strands. Then we have the piece of the genes on both the sides, these will be on the foreign DNA as well as the vector. So, vector will have an opening and the foreign DNA will have the similar structure, so that this will come and fit in the opening, and then again this is been reconstructed with treatment of ligase. Now, ligase joins, you can see here in this second step, the desired gene is been joined with the plasmid after being treated with ligase, and then we have a chimera or we have a recombinant plasmid. Then this plasmid is reintroduced into the host organism from where it was taken. Suppose the plasmid was taken from the bacterium E. coli, then it will be taken back again to E. coli, and then the E. coli is allowed to be cultured in artificial medium under control conditions. Okay. When they grow, we shall find out they are growing with the particular antibiotics that is having the antibiotic resistance, then we confirm that it has got transformed, then we are sure that the recombination has taken place. And once it is been done, we shall see that it is now ready. Then suppose we want to have a particular product from this particular gene we have introduced with, then we shall allow it to grow And we do the same with the bioreactors in huge scale, and once it is growing to a particular extent, in the meantime we shall be taking out the culture, examine for the expression of this particular one, and then we shall see that the product is ready, we shall take out the culture, then it will go for the downstream processing. In the downstream processing, we shall see this is nothing but chemical engineering part. We shall have control things, we shall purify the molecules we are interested for, and then these molecules will be purified, processed, and then they are ready for marketing. So, this can be vaccines, this can be medicines, this can be anything we are interested for. You see, we have been using this technology without understanding it for our benefit earlier. Now we are understanding this thing, we are using the technique in a newer way, so that we shall use it in a modern condition, and we get medicines, vaccines we can use for many things. This we shall be discussing in the next class. I hope you have understood the topic very well. In the next class, we shall be discussing more about this. By the time, if you develop any doubt, you have to go through your text where these are beautifully explained. You can go through animations from internet, you can understand it better. Thank you for a patient listening. Thank you all.